This is a recording from Early Career Perspectives on Future Earth, Ecology and Civilization, from the first session on the future of biodiversity in the Anthropocene, with Rob Sawara Gomez presenting on In Search of the Calyx of Eternal Youth, a biodemographic perspective.
As Peter indicated, I am an early career researcher myself. I'm based at the University of Oxford in the Department of Zoology. And I'm interested in what makes things go decrepit and what makes things avoid decrepitude. Examining the causes of aging has kept researchers busy for hundreds of years. In Greek mythology, Ambrosia was the nymph responsible for bringing a calyx full of nectar from the flowers of the plants with the same name. This elixir had divine properties, allowing gods and goddesses alike who drank from it to remain youthful. The search for the calyx of eternal youth, even though a poetic enterprise, has motivated researchers to examine the patterns and processes that promote the lengthening of lifespan in humans and other species populations. And that's the calyx from that word I was just referring to. It is precisely this that it consists one of the lines in the research in my group, the Zygote team at the University of Oxford, where we use a combination of biochemical, physiological, demographic, and also computational tools to explore which, where, and how some species evolve senescence while others don't. Now, senescence is defined as the physiological deterioration of the organism that ultimately results in a decline in fertility after maturation and an increase in the risk of mortality with age. The exploration of senescence is a rather old field itself, going back to even Aristotle, who in one of his treatises, he got to state that in order to live long and healthy, one should go for daily walks and restrain the amount of food. So he was already referring to one of the main theories of the avoidance of senescence, which is caloric restriction. Historically, there's been dozens and dozens of hypotheses for the evolution of senescence, how it comes about and how it persists. Nowadays, however, we usually test three predominant explanations. The first one, which was put together and forward by Medawar, is the so-called mutation accumulation. Nature is a highly competitive place where most individuals die before they can even become of age. Thus, why should we even invest on maintenance? Peter Medawar suggests that senescence comes about due to the imperfect maintenance functions in organisms, whereby mutations accumulate with age, and these mutations eventually become fatal for the whole organism. A second theory is the antagonistic pleiotrophic theory. Senescence is a two-side effect on necessary functions. Most genes, as we all now know, carry more than one function. Pleiotrophic genes specifically carry out a function that is beneficial early in life, such as an increase in reproduction, and that same gene may incur a cost late in life, such as an increase in mortality. The third theory, the so-called disposable soma, states that because the body allocates resources to metabolism, reproduction, repair, and maintenance, and because the resources are limited, the body, the organism, must compromise, and in doing so, it does not so optimally, and as a result, senescence emerges. Now, what we do know is that senescence has a genetic basis. The vast amount of work in age of research in the last decades has resulted in important breakthroughs. In the case of fruit flies, Drosophila melanogaster, for instance, we have come to realize and find out that there are some genes like the P53, which is an important mediator of longevity. Expression of the dominant negative DN form of this gene in adult neurons, but not in the muscles or fat of the body cells, expands the longevity of these species, resulting in a survivalship curve, as indicated by the black line, which can expand not just the longevity, but also the amount of individuals that get to advanced ages. On the flip side of the coin, we also know that the environment affects the rates of senescence. In a recent study in rhesus monkeys, it was found that a decrease in resources that was not associated with malnutrition, so-called caloric restriction, increased longevity and delayed the onset of age-associated disorders as well as other causes of mortality. Now, up until recently, the predominant tenet on age of research was that senescence was universal, or as Hamilton widely put it once in a published work, that senescence is in fact inevitable and will be found even in the farthest reaches of the universe. And he continued, and even in the most bizarre of life forms. What is the same to say that the pattern that we find here, where fertility after maturity in blue declines sharply and the risk of mortality in red increases sharply as well, should be found not only among humans but among monkeys, among whales, among horses, and even invading into other kingdoms like in plants. In a recent publication, my colleagues and I contested these tenets. We showed, using a comparative demographic perspective, that senescence not only is not universal, but it also seems to be an exception rather than the rule. Here we examine the performance of individuals for the risk of mortality in red and the fertility contributions in blue as individuals get older, from the age of first reproduction until 99% of the individuals in those populations decline. And we did so for 48 species, ranging from humans to other animals, and thrown into the mix also some plants and brown algae. These appeared organized in this framework from the top left for species with the greatest degree of senescence to the bottom right with those with a lower degree of senescence. Just to walk you through it, because this figure is a bit complicated, on the very top left we find a cohort of Japanese humans, and these are behaving according to the Hamiltonian expectation of senescence. That is, a sharp increase in mortality risk at advanced ages, which goes in the case of this population from 15 years old until 102, and a decline in fertility after the peak of reproduction. So is the case for other species, such as the guppies, the hunter-gatherer human populations, and the southern fulmar. An increase in mortality is also shown, and a decline in fertility. Now, as we walk down this panel, we can also start to see that for certain species, such as pines, crocodiles, and yellow-bellied marmots, an increase in fertility with age is shown. It is true that the mortality also increases with age. Last but not least, perhaps towards the very bottom on your right-hand side, you can see a range of different species belonging to different taxonomic affiliations. We've got some orweeds, we've got some oaks, and the famous desert tortoise, for which the rate of fertility plateaus at medium and advanced ages, and it is the rate of mortality that declines with age. When we contrast and compare the curves of survival and fertility for all of these species, one cannot ask but wonder, why have we been asking the question, why do humans senesce? And it is in fact this paper, I would argue, that has made a paradigm shift in the field of aging research. We should stop asking why do we senesce and start asking how come that other species don't senesce? How come that senescence is in fact more of an exception rather than the rule? In my group, we take a triple approach to examine the evolution of and the escape from senescence. This ranges from comparative biological analysis using big data to experimentations in the field using long-term demographic censuses, and also biochemical assays to underpin the mechanisms behind these patterns. Now, in the interest of time for today's webinar, I will only focus on the first part, which is looking at very general strokes of the patterns on the evolution of senescence. And to do so, I will be using data that comes from the Comparative Plant Matrix database and the Comparative Animal Matrix database. These are two open access resources that I have been leading at the Max Planck Institute for Demographic Research, and some of the undergraduates and graduate students involved in this project in my team in Rostock, Germany, can be seen on the screen. These two repositories include hundreds and hundreds of high-quality demographic data for plants and animals, and this enterprise has now become a multi-institutional enterprise, including not just the Max Planck, but also 10 other institutions. The data in these two repositories is truly global. It is true that although most of the demography comes from Europe and from the North Americas, we also have got high-resolution demographics for plants and for animals in other continents. And the access to these data now allow us to ask for the first time, what are the general patterns behind the rates of senescence that we observe in plants and animals across the world, the globe, and across the tree of life? What I'm trying to depict in this figure is to break rule number 1 of a presentation, which is thou shalt never put in a single slide more than 1 or maybe 2 panels. What you can see here is over 300 panels, which correspond to over 300 plant species from the compadre plant database. These are ranked, just like in the previous paper, from species on the top left that undergo high rates of senescence to species on the bottom right, which undergo low rates of senescence. And the reason that we color-coded this according to the growth forms is that we were interested in knowing whether the growth form was a good predictor of the rate of senescence. So because the colors are not aggregated in space, that means that in fact they are not. What was interesting from this line of research is that we found that 50 plant species in this data do undergo or evolve senescence, whereas more than 250, so that's the vast majority of the species that we have looked at, manage to somehow escape from senescence. So in them, individuals, as they get older, they become more reproductive and they diminish the risk of mortality with age as well. Now, in order to understand how they do so, we need to take into consideration how they allocate to different functions. And this is something that is pivotal to all three main theories on the evolution of senescence. And so to do this approach, what we did is we derived key life history traits, metrics of the time, intensity, and frequency of allocations. And you can see them on the slide. These have to do with investments on the next generation. These have to do with investments on the longevity of the individual itself, on 4 different moments of reproduction, and how individuals may grow and shrink as they get older as well. With this data, we also took into consideration the multidimensionality. And what you can see there is the space occupied by different taxonomic groups in the plant kingdom, according to this 10 life history traits framework. And we will be using this framework to try to predict what are the combinations of life history traits that result in escape from senescence. We also take into consideration the fact that the data in this analysis are not independent, so we are controlling for phylogeny. And we also, in the paper published in PNAS last year, looked at the effects that the environment and the anatomy had on the overall patterns that we found. The overall results can be summarized in a simple principal component analysis. The main amount of variance was explained by 2 PCAs, or 2 principal component axes, which added up to 60% of the variation. And the phylogenetic signal was rather low, implying that the repertoire of life history strategies we're seeing here is not constrained by the ancestry of the species. From left to right, we can see an axis that runs for individuals that grow fast versus live long and allocate lowly to generation time. And that's the so well-known fast-low continuum. From top to bottom, we can see the 4 different moments of reproduction, the intensity, frequency, timing, and duration on allocations to reproduction versus shrinkage. This is a novel axis that hadn't been reported before, and we refer to it as the reproductive strategies axis, absorbing 21% of the variation in the data. Now, in a latest paper published this year in New Phytologist, I used this framework with more species, in this case not 418, but 650, to test whether how much a species allocates to growing fast versus living long, or reproducing a lot versus having the ability of decreasing in size, would allow us to explain traits such as senescence. We know from this publication that if we know basic characteristics of the fast-low continuum and the reproductive strategy axis, we can in fact predict the rates of growth of the population. Red means good news for the populations that are increasing. Blue means not so good news for the populations. We also know that using these scores on the PCA axis, we can predict the degree of demographic resilience, with redder colors being populations of species that are more resilient to external perturbations. We now understand that species that live short or fast and species that have got a high probability of shrinking at the individual level have a high rate of resilience. Using this framework, I've also been able to couple it up with other open access repertoires, such as the Tri-Functional Trait Database, to explore how functional attributes of the plant, such as the specific leaf area, that is the ratio of the area to the dry mass, correlates with life history strategies. And in doing so, we've been able to find that species that live short produce cheap leaves, and that most species that live long produce very expensive, very low value SLA leaves. With the exception of the species on the top right corner, so those are species that live long and reproduce quite extensively. Now we'll come back to that in just a second. I've also been able to track down the importance of contributions of all individuals to a population by quantifying the force of selection on them. If the force of selection is high, that means that in those populations, all individuals are under strong selection and should therefore continue to contribute through high rates of survival and reproduction. Those are the species that we have typically found on the top right corner in yellow on the PCS space, corresponding to species that live long and are again highly reproductive. When we put this together with the understanding of how a species allocate to specific leaf area, how a species may escape or not from senescence, and their current conservation statuses, a really multidimensional, complex, and useful picture emerges. And that is that species on the top right, the species that manage to invest both highly on survival and on reproduction, happen to be the invasive species. Those are species that are more predominantly escaping from senescence. And at the same time, even though their counterparts of equal longevity allocate highly to specific leaf area, those species, the invasive species, do not. So what I would like to propose is that an integration of demography, of functional traits, and conservation biology may be the key to understanding invasions, as Martin Winter will also elaborate in the next talk. But of course, the audience may be wondering, and what about animals? Yes, we do have access to hundreds and hundreds of animal species with high quality demographic records through the Compadre Animal Matrix database. It turns out that the framework of life history trait decompositions on a principal component axis works just as well in animals as it worked in plants. Animals in this case are in blue and plants are in red. From the standardized demographic records, we can also measure when most of the mortality happens as a function of age. We use a measure of entropy to that end. We call that Kiffitz's entropy. When Kiffitz's entropy equal to 1, mortality is spread equally across all ages within the population. When it is lower than 1, it accumulates mostly at advanced ages, implying that the species senesces. And when it is greater than 1, most of the mortality happens early in life, and past a certain threshold, almost no mortality happens within the individuals of the population. We can then contrast the rates among animals in blue and plants in green, and find that in the case of animals, most of them do undergo senescence, but the range of rates of senescence in the plant kingdom is truly wildly broad, ranging from those that undergo senescence to those that do not. Now, we can also break it down by taxonomic affiliation, and what we find is that it is not quite true that animals systematically undergo senescence and plants escape from that. In fact, the picture is a bit more complex. We find that bony fish, birds, clams, insects, and most mammals are in a propensity towards the evolution of senescence, whereas other animals like corals and the indeterminately growing reptiles tend to be more like plant-like, demographically behaved asenescent organisms. So going back to Ambrosia, the nymph that was able to keep the juvenility of the gods and goddesses in heaven, it may be the case that somehow corals, reptiles, and some plants might have found the calyx of eternal youth. Now, what's interesting is that most of the demographic research being allocated to agent research is not done in plants. It's in fact done in humans for obvious reasons and in human-like organisms like monkeys. It's interesting, I say, because in the backdrop we have got a really amenable system that is of plants that, as famously put by the founding father of modern plant demography, John Harper, are just waiting outside to be counted. And counted is, in fact, the fine grain in the daily basis of demographic research. What I would argue going forward we need to do is to, having understood that there's a really broad landscape on the rates of senescence, we need to start looking for the patterns behind this very complicated picture. We now know that senescence is not universal. We also know that allocations to trade-off between growth, survival, and longevity are not as strict as we thought they would, or as strict as classical works in life history theory told us they would be. We know that those species that somehow bypass the trade-offs to longevity and reproduction are in a high likelihood of escaping from senescence. Caloric restriction is a really hotly debated topic, and I think that more research should be allocated to testing that hypothesis, not just within the animal kingdom, but also among other taxonomic affiliations such as plants and fungi. And the study of demographic oddities, that is, animals that demographically behave like plants, like corals, and plants that demographically behave like animals, like carnivorous plant species or mistletoes, which are effectively parasitic of other species. Those are where I think the keys to the understanding of senescence may be found. So I would argue that getting to the roots, both in a philosophical and in a literal taxonomic way, is fundamental to understanding the broad diversity on the rates of senescence. Some of this research that I have highlighted can also be found in a recent publication that my colleague Rich Jefferson, Owen Jones, and myself published this year. With that, I would like to conclude by thanking the audience, thanking the organizers, and also my funding agencies. Thanks very much.
Thanks for watching this presentation from the Early Career Perspectives on Future Earth conference. You can see the next talk in the conference highlighted here on the slide, and we hope you will enjoy many of the other recordings. Thank you.